In this video, we will begin to analyze the components and important aspects of viral replication. We will begin with the bacteriophage lytic cycle and bacteriophage lysogeny. Viruses are obligate intracellular parasites. They are dependent upon their host for replication because they utilize the host organelles and enzymes to produce more virions. The viral nucleic acid contains only a few genes. The genes of viral nucleic acids are responsible for the production of structural components such as capsids and glycoprotein spikes, and for a few enzymes required to complete the viral life cycle. These genes are transcribed and translated by the host cell's enzymes and ribosomes. Thus, viruses can only replicate when they gain access to a host cell. Once they gain access, the activities of the cell are redirected so that the host cell becomes devoted to synthesizing viruses. We will be examining the replication cycles of bacteriophages and animal viruses. We will begin to explore replication cycles with the lytic replication of bacteriophages. Lytic replication cycles end in the lysis and death of the host. Replication cycles may also be referred to as infectious cycles. Infectious cycles include all of the activities from the time the virus contacts the cell until the end when new virus particles have been synthesized and released. Virologists divide the replication cycles into steps to facilitate their study, but there are no such boundaries truly occurring. Lytic replication in bacteriophages encompasses 5 stages: attachment, entry, synthesis, assembly, and release. The bacteriophage observed in the image is a T4 phage that infects E. coli cells. T4 phages are a type of T-even bacteriophage. T-even bacteriophages are large, complex bacterial viruses with a characteristic structure. The T4 virion in the image is composed of an icosahedron head that houses the phage double-stranded DNA and a helical tail sheath to which a base plate and tail fibers are attached. The first stage of replication required for the T4 to infect its E. coli host cell is attachment. After randomly colliding into the bacterial cell, the bacteriophage attaches to the bacterial cell. Attachment is based on the tail fibers of the phage being complementary to receptor proteins on the surface of the bacterial cell wall. The specificity of the tail fibers for a specific chemical receptor on the surface of the bacterial cell ensures that bacteriophages only attach to specific species. Bacteriophages may attach to receptor proteins located on bacterial cell walls, flagella, or pili. After attaching to the bacterial cell, the phage injects its DNA into its host. This step is known as entry or penetration. In order to carry out this process, the phage releases an enzyme known as lysozyme. Lysozyme degrades part of the cell wall. The tail sheath of the phage is observed to contract as the phage nucleic acid is injected into the cell. Within the sheath is a hollow tube that penetrates the cell wall where it was weakened with lysozyme and then the cell membrane. This allows the nucleic acid to move from the icosahedral head through the hollow tube into the cell. The action is very similar to that of a hypodermic syringe. Following entry, the nucleic acid resides within the host cell and the capsid remains outside. After the viral DNA has entered the cell, viral nucleic acid directs the degradation of the bacterial chromosome and causes host protein synthesis to cease. The cell then begins using its own enzymes and nucleotides to build copies of the viral DNA. Soon after the bacteria has started creating viral nucleic acids, the cell begins to synthesize mRNA from viral genes. Translation of the viral mRNA is carried out by bacterial ribosomes to produce viral proteins, including the capsomeres of the head and tail. Toward the end of the synthesis process, the cell begins to produce lysozyme, which acts to weaken the cell wall, allowing the virus to escape once they have been assembled. Following synthesis, a growing pool of viral components begin to accumulate within the bacterial cytoplasm. The viral components begin to spontaneously assemble to form virus particles. The nucleic acid is assembled into the icosahedron head, while the tail components assemble separately. The tails spontaneously attach to the head to form the virion. During this process, some icosahedral heads accidentally enclose pieces of the bacterial chromosome that were still present in the cytoplasm. This bacteriophage is not infectious but can still bind with the host cell and transfer bacterial DNA to another cell. This process is known as transduction. The final process is the release of mature bacteriophages from the bacterial cell. This is accomplished as the lysozyme finishes degrading the cell wall, allowing the cell to lyse, thereby releasing the bacteriophages. The replication process for the T4 bacteriophage requires about 25 minutes and is able to produce approximately 100 to 200 new bacteriophages. Bacteriophages, known as temperate phages, may enter a cell and not result in the lysis of the host bacterial cell. Temperate phages must attach and enter into the host cell just as the T-even phages in the lytic cycle. However, after entry, the temperate phage does not degrade the bacterial chromosome. Instead, the viral nucleic acid is incorporated into the host chromosome. The viral nucleic acid is referred to as a prophage when it is incorporated into the bacterial chromosome. The majority of the prophage genes are repressed by two genes that are transcribed and translated into proteins. The proteins keep the cell from synthesizing viral components. When a bacterial cell that is infected with a temperate phage undergoes binary fission, the viral nucleic acid is copied, and those copies are found in the daughter cells. Thus, the prophage may remain latent in the progeny cells as well. Lysogenic phages may be triggered to enter into the lytic cycle. Exposure of the host cell to UV light or certain chemicals can cause the virus to be removed from the bacterial chromosome, at which time the virus enters into the lytic cycle. This process is known as induction. Following induction, the viral nucleic acid will direct the degradation of the bacterial chromosome. The steps seen in the lytic cycle are followed to synthesize, assemble, and release new virions. Bacterial cells that are infected with a lysogenic virus are not able to be infected by the same type of phage, so cells are immune to reinfection. Bacterial cells harboring a prophage may also undergo lysogenic conversion. Lysogenic conversion is also referred to as phage conversion. Lysogenic conversion results in new phenotypes for bacterial cells that are infected. In some cases of lysogenic conversion, a harmless bacterial cell is able to become a pathogen. In many cases, the prophage genes permit the bacterial cells to produce toxins, as in the case of pathogenic forms of E. coli, Clostridium botulinum, and Corynebacterium diphtheriae.